Greeting to my supervisor, Associate Professor Dr. Hairul Azman bin Roslan and fellow examiner. My name is Umi Umairah binti Muhammad Syukur and my magic number is 72719. Today, I will share with you my proposal project with the title The Genomic Transformation of Mung Bean with Sago Palm Chitinase 2 cDNA. In this presentation, we will cover the introduction, objective, literature review, material and method, expected outcome, working schedule, and references. We start with introduction. The research aimed to transform chitinase 2 cDNA from sago palm, Mactoxylon sago, into mung bean without radiata to counter fungal infection. The chitinase is the hydrolytic enzyme in the plant that hydrolyzes chitin polysaccharide located in the fungi cell wall. While we use mung bean used as a plant model to test the expression of chitinase 2, whether the chitinase is able to protect the mung bean from fungal infection. Mung bean is chosen to be plant model because of its small genome size, easy to culture, and simple for genetic manipulation. The small genome size enables it to express the chitinase 2 cDNA, producing plant transgenic. Chitinase 2 used to be transformed into Bigna radiata by application of agrobacterium-mediated transformation method through quaternary non-explant. The chitinase 2 cDNA is isolated from XL1 blue before transformed into PGSA1131 plasmid vector and followed by transferring into cloning vector agrobacterium tumefaciens and continue to transforming into the host cell mung bean. Why do we need to conduct this experiment? It is because fungal infection generate crop disease, damaging crop organ and stimulating biotic stress. Moreover, fungal infection outbreak cause a global reduction of crop yield, and lastly, fungal infection decrease the quality of crop product, leading to the food safety risk. Therefore, transforming the chitinase 2 from M. sago into B. radiata will produce recombinant plant with fungal resistant. In the end of the research, this experiment will answer whether chitinase 2 is able to be expressed in the transformed mung bean to reduce fungal infection. This study also needs to identify the difference between sequencing and molecular characterization of mung bean before and after carrying the chitinase 2 cDNA. The objectives for the research is to transform the chitinase 2 cDNA into mung bean by agrobacterium-mediated transformation method. Secondly, to identify chitinase 2 cDNA expression in the mung bean genetic sequence, and thirdly, to differentiate the molecular sequencing and characterization in mung bean before and after transformation. We move to the literature review. They explain about fungal infection of crop in type of fungal disease. Berlanga state that powdery mildew derived from the Acidiferous fungi family affected the greenhouse-grown cucumber with a threshold for economic damage at 20% infected leaf area. As well as the Gaskell in 2040 explained about Plasmopara hastidi in the fungi that causes sunflower downy mildew disease. The symptoms appear when plant droughing, leaf bleaching at the beginning of the veins, and appearance of white sporulation. In explaining the impact of fungal infection on crop, Fisher in 2012 proved that fungal infection reduced the crop yield. Yersinia graminis attacked the wheat, resulting in crop loss up to 70%, while Godfrey claimed that the animals' losses of crop due to the fungal infection increased the food crop price. Godfrey predicted that international commodity prices of rice would equalize each year, and the rising demand for rice from the impacted nation would boost global rice prices. Means that the extreme reduction of rice product because of fungal disease epidemics causing the price of the rice to rise. Moving to chitinase, Hamid and Ahmad found that the role of chitinase in plant are generated as pathogenesis-related protein in response to phytophytogen infection or reaction to elicitors such as chito-oligosaccharide or growth regulators such as ethylene. An example of research about chitinase as a fungal-resistant crop was done by Kumar in 2018, in which he applied oligomeric chitin compound produced from chitinase to enhance plant defense and growth for fungal resistance. For the plant model system, Hassan pointed out that mung bean is used as a plant system because it has a rapid life cycle, self-pollinating, and tiny genetic gap compared to other legumes family plants. The seed germination of mung bean only takes 3 days if cultured in the media. A study by Kang in 2040 claims that mung bean have short genome size. The study found that the genome size of mung beans is only 579 megabases. Next, The example of research used mung bean as a plant model is done by Jaiwal in 2001, which he applied explant of primary leaf and cotyledonary knot as well as the binary vector containing reporter gene Cas and their selection marker MPT2. Jaiwal also used mung bean as model plant in his 2017 research, in which he transformed the code A gene into mung bean for abiotic tolerance. Moving on methodology. The first method is culturing XL1 blue competent cell carrying chitinase 2. XL1 blue is cultured in the LB agar containing epicellin. Secondly, extraction of chitinase 2 from XL1 blue plasmid vector. The isolation of chitinase 2 from XL1 blue needs to be done first. In this method, LB pros containing XL1 blue is spun in the microcentrifuge, resuspend, incubate for 1 hour to remove the cell wall debris. Then, add the PCI and isopropanol and wash with 70% ethanol to precipitate the nucleic acid. Then, followed by analysis of agarogel electrophoresis AGE and amplification of chitinase 2 cDNA by PCR. For the PCR, add the tape polymerase, distilled water, primer 1 and 2, lastly chitinase 2 cDNA template. Then, the quantification of spectrophotometer will be done to calculate the amount of chitinase 2 present. After isolation of chitinase 2 from XL1 blue is successful, the chitinase is transferred into PJSA1131 plasmid vector. The medium is grown for 24 hours at room temperature until colonies of E. coli containing PJSA1131 growth. After transformation of chitinase 2 cDNA into PJSA1131 plasmid vector is continued with the PCR diagnostic to identify the presence of the genomic DNA. The next step is transformation of chitinase 2 into mung bean by agrobacterium mediated transformation method AMT. The method starts with preparation of mung bean seed. The seed needs to be sterilized, soaked in mercury chloride and cultured for 5 days. For half-stick explant, we need to decoating the seeds and carefully remove one of the cotyledons. Continue with preparation of agrobacterium tumefaciens competent cell culture. The agrobacterium tumefaciens is cultured in the YMB media. The antibiotic that need to be added are kanamycin, streptomycins, and rifampicins, followed by agrobacterium freeze-thaw transformation using PGSA1131. Plasmid DNA is introduced to competent cell, then incubated on ice and heat shock in 37 degrees Celsius in water bath. Then we incubate in the spinning shaker after addition of culture into LA plate, followed by DNA plasmid extraction and PCR to validate the existence of transformed cells. Lastly, integration of chitinase 2 cDNA into mung beans. After seed germination of mung bean, slice horizontally the cotyledon and pour the agrobacterium culture into the wounded cotyledon. Then, analyze the presence of chitinase 2 in the mung bean by PCR. Finally, for result analysis, it will be done by characterizations and molecular sequencing. The genomic DNA is first isolated using CTAC technique, followed by PCR, then undergo Southern blot analysis. The blast analysis is used to compare DNA RNA between transformed and non-transformed mung bean that function as the controller. The expected outcome for this study is mung bean is expected to express the chitinase 2 cDNA successfully by application of agrobacterium mediated transformation method. Furthermore, the expression of chitinase 2 in the mung bean predicted to be able to be identified using Southern blot hybridization analysis. Finally, blast analysis of genetic sequence in transformed mung bean is expected to be different from non-transformed mung bean due to the integration of the chitinase 2 cDNA. Next is work schedule. Based on the Gantt chart, from October and November 2021 is for proposal writing, followed by proposal presentation in January 2022. For final year project 2, extraction of chitinase 2 from XL1 blue, preparation of agrobacterium tumefaciens, and transformation of chitinase 2 into mung bean is predicted to be done between January and March 2022. The final year project is expected to complete in June 2022 after FYP presentation is done. References. This is the attachment of the references that I use for the slide proposal presentations. That's all for me. Thank you for listening.